All right, good evening, everybody. Thank you all for coming, especially exciting to see some incoming MCRS students in this room too. Glad that you guys could make it. My name is Michaela Coates. My teammates with this project are Kai Dang and Carmen Gardner, and today we will be presenting our assessment of habitat suitability in Orange County for the endangered black abalone. Like Sarah said, our community partners with this project have been employees of NOAA Fisheries, which is the federal agency that manages marine endangered species. They're also on the Black Abalone Recovery Team, which is a collaborative group working towards black abalone research and recovery. Today, we will be talking about how black abalone came to be endangered, the steps that are being taken towards the species recovery, our 10-factor habitat assessment methodology, the results of those assessments, and our final recommendations for the future of black abalone recovery in Orange County. So to start, black abalone are large marine gastropods that live between the lower and the middle rocky intertidal zones. And now these two photos were very specifically chosen for this slide as they provide a good indication of what makes good black abalone habitat. So first, they prefer complex geomorphology. You can see them nestled here in the cracks and crevices and undersides of boulders that offer them protection. They also rely on species such as crustose-coralline algae, which is what you see there in pink, as it releases chemical cues that help support larval recruitment. And although not seen in these photos, they do rely on kelp as a food resource. They seem to have a few favorite species, but giant kelp seems to be their preference. And although that species does not grow intertidally, it does drift in from offshore kelp beds. Now, they currently range from Point Arenas, California to Bahia Tortugas, Mexico, which is the current range here seen in green. The extent of their range contraction or their historic range can be seen in red, and that red box is our study area of Orange County, California. Now, as you can see on the photo on the left, black abalone used to be extremely abundant in this region. However, that is no longer the case due to a combination of overfishing and, most significantly, the spread of a disease called withering syndrome. Now, this disease was first observed in the 1980s, and it affects the species' digestive organs and atrophies its muscular foot, which you can see in the picture on the right, and that leads to mortality through starvation and or predation. And as such, they experience declines of up to 99% throughout portions of their range. Now, with the loss of a species also comes the loss of their ecological and cultural roles. As an herbivorous consumer, they influence community structure through the maintenance of substrata. Decreases in abalone abundance have been found to be correlated with decreases in percent cover of crustose-coralline algae, as well as bare rock, and increases in percent cover of sessile invertebrates, which not only creates a barrier to their own recovery, but limits further recruitment of other species. They were also an important cultural and financial resource, not only for the indigenous communities of the West Coast, for diving communities, and the valuable commercial fishery, which sadly closed in 1993. Now, they were listed as federally endangered on the Endangered Species Act in 2009, and 2 years following, critical habitat was designated, and just last year in 2020, their final recovery plan was published. And now this recovery plan outlines 8 major recovery actions, where Action 3 focuses on supporting natural recovery and enhancing local populations through methods such as captive breeding and outplanting, and Action 5 focuses on protecting and restoring their habitat. Now, when critical habitat was being designated, the Orange County coastline was excluded in part due to their local extinction. However, recent population surveys have found a handful of individuals, and the combination of historical occupation and the potential for success of restoration and transplantation lead us to believe that this region could still support black abalone recovery. However, habitat surveys in this region have focused solely on geomorphology, so the knowledge gap that we are filling is a comprehensive understanding of habitat suitability of rocky intertidal sites and population status in Orange County, California.
So we addressed 3 research questions. 1, we answered which intertidal sites along the Orange County coast provide suitable habitat for black abalone. The corresponding objective included conducting habitat surveys, human impact surveys, and assessing the current black abalone population. Next, we answered which sites have the highest rates of human visitation and impactful behaviors. To examine this, we developed a metric for assessing potential human impacts at survey sites. Finally, we found which sites have the most abundant and persistent offshore kelp biomass. We found this by examining kelp bed biomass abundance, variability, and persistence offshore using remote sensing. We assessed several habitat factors. We assessed geomorphology, which is the complexity of the rock structure at each site, and assessed the total surface area and quality of habitat. We assessed biotic interactions, which include interspecific interactions, food availability, and predators. And lastly, we assessed human visitation to the habitat and human behaviors at each site. We first followed the UC Santa Cruz established Black Abalone protocol, which is used across the state to assess geomorphology. Last year, MCRS students built on this protocol by including belt transects. We estimated the percent of good, moderate, and poor habitat. We assessed substrate relief, sediment, and exposure. We used a random number generator to get 10 points along a 50-meter transect to measure the area of cracks and crevices and area of boulders within perpendicular 5-meter transects at each random point. For biotic interactions, we estimated the percent cover of biotic organisms in the 5 best cracks and crevices surveyed. We completed 30-minute time searches at each segment for predators and measured sea star predators from the center of the disc to the tip of the longest ray with a caliper. We surveyed the entire segment for presence or absence of macroalgae and noted if it was in situ or drift. To further assess food availability, we analyzed abundance, variability, and long-term persistence of giant kelp biomass remote sensing data from 1984 to 2020 provided by Dr. Tom Bell of UCSB. We created 10 2x2 kilometer boundaries to extract the time series data from these specific locations shown on the map. We assessed abundance by looking at the current and historical means. Variability was assessed across sites by analyzing coefficient of variation and consecutive disparity. And long-term persistence was analyzed by looking at the number of years with zero kelp. To assess human impacts, we looked at human visitation to the specific abalone habitat as the number of people per site in the area during a 2-hour survey. The sites along the Orange County coastline range from south of Newport Harbor down to Dana Point Harbor. We recorded the MPA status as our sites were located within 3 different MPAs, which are shown on the map. We then analyzed citizen science data collected on human interactions during a walking survey that was provided as MPA watch data from the OC Coastkeeper. Here are 2 maps of our 18 habitat assessment segments within sites that are shown with an aerial imagery background. If you refer to Gough Island and Treasure Island in the bottom left corner, the aqua-colored polygons are the habitat segments where we completed surveys. The light orange buffer shows the approximate range we created to assess human visitation.
Right. So this is our scoring system. So we survey 10 factors for each site, and then we assign whether the site has a positive, neutral, or negative for each factor. So the factors in the white rows are assigned by categorical criteria, and factors in the blue rows are scored assigned by quartile values. So we have all the data from that site, from all of the sites, and then we calculated the quartile value so that we can assign a score for them. So before we jump into our resource sections, we just want to recap a little bit here. So these are all the factors that we assessed, but we want to focus more on the human impact factors and the offshore kelp factor, because those are additional factors that we built up from last year project. So for our human impact data, on the left side, you will see all the sites in geographic order from north to south. We have the red violation rate, and the green is the informant's present rate. So we did some t-tests, and the results show that there is no significant difference between enforcement presence in SMCA and SMR MPA type, but there is a significantly higher violation rate in SMCA than SMR, and there is a weak correlation between the violation rate and enforcement rate in all MPA type. So this is the graph of moving average cow biomass from 1984 to 2020. So we calculated moving average by taking 5 years interval. We have average from 1 to 5, and then from year 2 to 6, and year 3 to 7, and so on, to get this map. And so if you look at the map, you will see the curves represent the biomass. So the higher the curves, the higher abundance. And then we have a flat area between 1995 to 2000. So that area is a long period of low biomass. So that represents low persistence. And if you look at the fluctuation of the map, the more fluctuating it is, the more variable it will be. We also added some time mark to show some significant period. So the red represent marking El Nino years. So you will see significant drop of kelp after an El Nino years. And that's why we look at the last 5 years after the last strong El Nino years to evaluate our current kelp biomass. We also mark the year from 2002 to 2012. These are the time period for Orange County kelp restoration project. And these are some critical value that we calculated for kelp in Orange County. So the first 2 columns are abundance. So you want to look for higher value. The third and fourth are variations. So the lower the numbers, the less variations, the better the kelp situation. For the last 2, we have a persistence. So we also want to look for lower numbers because lower numbers mean higher persistence and better kelp conditions. And overall, we have better kelp conditions in South Orange County than North Orange County. So this is the result of our habitat assessment. So if you look at the top 3 rows for UCSC protocol, MCRS transect, and the boulders, these are the geomorphology data. So overall, we have a wide range of habitat quality in terms of geomorphology. So we have some good habitat, and then we have some bad habitat. So that is something to be considered. And for intertidal kelp overall, we pretty much have food for black abalone in every site, but we want to consider the condition of offshore kelp bed as well, because better offshore kelp bed will have higher chance of drifting kelp to the intertidal zones. And in Orange County, in general, we have quite decent interspecific interactions. So there are only a few sites that have negative interspecific interactions. And in most sites, there is very little predators present as well. So in terms of like neighborhoods, we have quite a good neighborhood for black abalone. For human presence and human behaviors, we consider these as the most impactful factors. but we can mitigate this. And for Black Abalone restoration project, we want to consider MBA status, but there's not much that we can do about these either.
So like Kai said, we took all of those factors into account, but we also took the idea of whether or not that factor could be mitigated or not into our final recommendations. So we recommend that sites SE2, GI1, DP2, and MC1 be prioritized as they all contain complex geomorphology, positive biotic interactions, including intertidal and availability, and human impacts were generally minimal, but those could also be additionally mitigated. However, we do not prioritize, do not recommend prioritizing CCSP2, WC1, or either of the CDM sites as they have comparatively poor geomorphology, as well as poor biotic interactions, including higher proportions of habitat degrading organisms that would ultimately be less efficient to restore, especially when coupling those sites with weak offshore kelp availability and extremely high rates of human impacts, including direct MPA violations. Now, our assessment continued to build on the novel protocol that was developed by last year's MCRS team. It overall took about 3 people, 2 to 3 hours to complete the fieldwork component of this study, but that ultimately provided us with a more comprehensive and quantitative approach to measuring habitat quality for Black abalone. So therefore, this new assessment protocol could be used to assess other sites or regions for the recovery of Black abalone, and our results can be used to inform the future recovery of Black abalone in Orange County. We wanted to give a special thank you to Amy Henry and our NOAA partners, Susan Wang, Melissa Newman, and Dave Whitting for their advisement during this project, and also all of the many entities that provided us with permits and site access so we could complete our habitat assessments. Another thank you to Dr. Tom Bell and CalpWatch, as well as OC Coastkeeper for the contribution of your data, which strengthened our analysis, as well as the many people who contributed to our project through fieldwork and conversations. Thank you all for listening.
Okay, thanks so much, Michaela. And now, if any of you have questions for Michaela, Carmen, and Kai, we have a couple minutes for them to answer those questions.
So I see in the chat, so the negative in MPA status just mean it has the least restriction. So compared to SMR, it has the most restriction, and then we have SMCA has just the least restriction. It doesn't mean like a negative effect necessarily, but like it's just the least restriction.
I'll ask a question. I'm curious about the progress and barriers to captively breeding the abalone for reintroduction, if you do find suitable habitat, and if you have anything to say about that.
The last update that we saw is Black abalone were not faring quite as well in the captive breeding process as white abalone were. As of last summer, I believe they had successful spawning, which is good, but there's still definitely progress to be made in the sense of captive breeding.
And there was someone raised their hand. I think that I saw Bobby's iPhone. Maybe we can let Bobby ask the question.
Yes, thank you to the Abalone team. I would like to know, perhaps in the future directions, if this project is continued, do you find that there may be opportunities for subtitle sampling that could help further our understanding of these habitat assessments? I know that typically abalone are found in intertidal zones, but I know there's some abalone species that can be found just below the surface subtidally.
Yeah, that's a good question. For the sense of black abalone, I mean, you're correct. They do stay in more of the middle to lower intertidal zone, but I think looking at this region subtidally would also be very interesting regarding kind of fluctuations that we've seen even in like green abalone populations in this region.
Great, thank you.
Okay, so let's give another set of funny clapping hand emojis to the Black Abalone team.